So good afternoon, everyone. Welcome to Amsterdam and the start of the ESDR. We're all very excited for this meeting, which is one of our first scientific meetings face-to-face. My name is Nick Reynolds, and on behalf of my co-chair, Emma Gutman, I'd like to welcome you here for this very exciting program today. So we've got a mixed format. Some of the talks are pre-recorded because of some COVID restrictions and other confounding factors. So it's my pleasure, first of all, to introduce Robert Bissonnette, who is the president of the IEC. He's got a strong track record in clinical trials and indeed is president of Innovaderm, which is a CRO specializing in conducting clinical trials with a particular interest in eczema, and he's obviously published very widely in the field. So I'm going to play his video and then we'll move on to the rest of the program.
Good afternoon and welcome to this IEC Symposium on single-cell technologies applicable to atopic eczema. My name is Robert Bissonnette. I'm the current IEC president. Unfortunately, I cannot be with you today. One important housekeeping comment is that this session is being recorded for later use on demand on our website. For those of you who may not be familiar with IEC, IEC is a non-for-profit organization led by experts on atopic dermatitis. We currently have 125 atopic dermatitis experts based in 28 different countries across 5 regions of the globe. IEC receives funding from various sources, specifically from corporate sponsors, and this is a list of our current sponsors. IEC is a very active organization. In addition to a board of directors, an executive committee, we have a nominating committee, an education committee, a membership committee, a finance and audit committee, and a research committee. If you're interested to participate, if you're a member of IEC and you would like to be active in one of those committees, please let us know. This is a list of people who are members of the Education Committee. This committee is extremely important because it makes symposia like this one possible. I want to thank specifically Nick Reynolds and Valeria Aoki, who are co-chairs of this committee. IEC Board of Directors. This is a list of all our current directors. I want to focus your attention on a few names. Alan Irvine, our president-elect. Emma Gutman, our immediate past president and one of the 2 IEC co-founders. Amy Powler, the other IEC co-founders. And Nick Reynolds, who is co-chairing the session today with Emma Gutman. IEC has been very active in 2021, mostly virtually, as most organizations, because of COVID-19, our activities were mostly virtual. We had our first in-person hybrid meeting in New York in 2021 at the ISDS meeting, and the topic was alopecia areata and atopic dermatitis. We have been very active this year. With meetings at the American Academy of Dermatology, our first symposia in Latin America, at the Bradla meeting. We were present at SID with a very well-attended session on microbiome, EADV, customizing the treatment of AD for diverse populations, and ESDR today. We launched a new program last year in 2021. Those of you who are not aware, we launched this program in order to help dermatologists and allergists to have a strong interest in ED and who live and practice in lower-income countries. People who would like to attend EAD, EDV, World Congress in Dermatology, specifically if they're presenting something in ED, but this is not an absolute requirement. And we offer reimbursements up to $3,000. We've also initiated clinical and research fellowships last year, and we have 2 new clinical fellowships that we've announced this year. Dr. Wang from Taiwan will work with Dr. Heigensfeld, and we have Dr. Mann from Germany who will work with Dr. Amy Powell. We've also changed our rules recently in order to expand IEC to welcome allergists. So allergists are now eligible to become members, counselors, or associates of IEC, like dermatologists. Now, our two co-chairs today, as I mentioned, Dr. Reynolds and Dr. Gutman, and I will let them introduce to you the very interesting agenda of today's meeting. Thank you.
So we're very fortunate today to have some outstanding speakers who are real experts in this rapidly developing field. Spatial transcriptomics can really be traced back to RNA scopes when people have started to visualize the expression of RNA in tissue. But with the advent of single-cell technologies, researchers around the world decided it would be fundamentally important to try and visualize the spatial context of these data expressions. And after several years' development, a group in Sweden published in Science in 2016 the first real methodology to take this forward. Since then, there's been a variety of different methods developed, and we'll undoubtedly hear some of these. But fundamentally, that technology involves, on a slide, permeabilizing the tissue, and then by gravity, the RNA goes through the tissue onto the pre-coated plate where there are oligos with barcodes, and that enables then, following reverse transcription, for these data to be spatially encoded back to where they came from, not quite yet at single-cell resolution, but still provided very important information about cell-cell interactions and other important developments. So this is what we're going to focus on today. And recognizing the importance of this development, Nature Methods awarded the Method of the Year back in 2020. And for those of you interested in the history and the kind of critical appraisal of the technology, I'd recommend this recent review published in Nature Methods. And this also includes a kind of indication of the number of publications in the field that is kind of growing exponentially. And I'm sure as we hear the exciting developments through our speakers today, this is a technique that's going to be very important to dermatology and atopic eczema in particular. So we'll introduce every speaker as we come to them fairly briefly, because there were further details of their biographies in the information that's been handed out to you. So it's my pleasure, first of all, to introduce Ms. Hanifa, who works dually between Newcastle, where she's Professor of Dermatology and Immunology, and also at the Sanger Institute as a Wellcome Senior Clinical Research Fellow and Faculty in Cellular Genetics. Ms. has been awarded a number of prizes. She's a member of the Academy of Medical Science and was a recipient of the Academy of Medical Sciences Folks Foundation Medal. And she's a leading member of the Human Cell Atlas Group, which is dedicated to taking forward single-cell genomics and making these data widely available for researchers. So her talk is again pre-recorded, so I'm going to play that now.
Thank you very much for the invitation to present an overview on single-cell genomics technologies at the IEC meeting at the ESDR. And thank you, Nick and Emma, for chairing this session. I'm going to go through very broadly the approaches in single-cell technologies and what it can teach us, and by illustrating some specific examples, and the next phase, and with regards to what we can do with the data. So the first slide, is something that a lot of people show in the sense of, what has single-cell methodologies enabled us to do? And this is essentially looking at the skin, for example, healthy or diseased skin like that of a patient with eczema, which has lots and lots of different cells in there. Keratinocytes, immune cells, and so on and so forth. And the conventional way of analyzing has been, particularly for genomics, has been to, essentially create smoothies, either all of the sort of fruit salad or composition of the fruit salad blended into one, or sort of specific, cell types taken out, for example, immune cells or keratinocytes, and analyzed by RNA sequencing. There are various other methods of analysis, but I'm going to focusing on the genomic sequencing, next generation sequencing technologies. But, in recent years, this approach has kind of like really scaled up in terms of resolution, in the sense that we can now deconstruct the tissue to its individual cellular constituents and analyze the whole transcriptome profile of those individual cells, much in the same way as if you were to take this bowl of fruit salad and deconstruct it, so that you can then assess what are the composition of the fruit salad and how many of those individual pieces make up the fruit salad. So this is very much a deconstruction approach, which gives you senses and also allows you to sort of look at cell states by RNA expression, and allows you to sort of give them cell labels or taxonomy exercise. And the deconstruction approach requires you to dissociate the tissue so that you get the individual cells in suspension. And in the early phase of the application of these technologies, you then use this deconstructed information from single-cell RNA sequencing, for example, to then reconstruct where the cells might be and which cells they may be interacting with. And this is very much done computationally using algorithms, so that you can then say keratinocytes are more likely to interact with Langerhans cells and so on and so forth, based on legacy knowledge, and look at receptors and ligands that are expressed by these cells on their surfaces to infer what the conversation may entail. But the technology has moved on, and now we can actually look at the tissue in situ in its intact form by taking tissue sections, and you can then look and perform in situ genomics or multi-omics. And these platforms rely on either hybridization using probes to RNA or protein, or targeted sequencing, for example, where you have a targeted probe which binds and sequences a particular transcript, or poly-A capture, which allows you to look at the whole transcriptome of specific tissue sites. And by barcoding the individual areas or imaging the tissue, you can then locate the RNA expression to those to the particular individual tissue site. However, there are many challenges of doing this. Firstly, in terms of resolution, whether you can actually measure the information at single cells. Most of the kind of hybridization and in situ sequencing methods provide the single cell information, because you can actually image the cell or actually analyze it for the specific cell boundaries. Some other techniques whereby you capture a particular area, which can be in the range of 10 microns to 50 microns, for example, will require you to then infer what the composition of those individual capture areas are. So that's one thing, whether or not the information is at, a size of area captured or at single cell resolution. And the second is the depth of what can be captured. Many of the hybridization and in situ sequencing methods enable you to measure hundreds or maybe up to 1,000 transcripts, but whole genome analysis at single cell resolution is still has its challenges and requires more computational approaches. The third problem with in situ genomics or multi-omics is scale, in terms of what's the size that you can measure. And that can range from, areas that are in the range of microns to millimeters. And you're now looking at whether or not you can analyze whole organs, whole organisms. So these are very different challenges, and the different platforms that are around may favor one form of either they're very good at scale or resolution and not so good at depth, or better at depth but not so good at resolution. So what can you actually measure using single-cell multi-omics? When the technology first developed, particularly when you were looking at suspension analysis, much of the measurement was about RNA, so basically the entire transcriptome of the cell, and that was what you could measure. But now a lot of platforms allow you to measure more than one parameter, so you can combine RNA measurement with, for example, surface protein, looking at site-seq technology, for example, or combining it with the open chromatin measuring epigenome. And this is, the single-cell multi-ohm type platforms where you can measure open chromatin and RNA simultaneously. One question that you could ask is, which is a truer measurement of a cell? Should you be relying on RNA, or is protein more relevant than RNA? Or what about the epigenome? And in some ways, I rather like to look at it as measuring different aspects of the cell. They're all complementary and none is more faithful than another. So you could imagine it as if you were to have a photograph of a person compared to an MRI scan, compared to a thermal scan, or CT scan. They give you very different information, but it's all very complementary information and gives you a lot more understanding about cell. There is also a global initiative which I've been involved in, and that's leveraging a lot of single-cell technologies to really profile the 37 trillion cells of the human body. Anyone can join, and I've just put the website there. And at the moment, there are researchers from more than 80 countries, more than 1,000 institutions, and more than 2,000 researchers. And this is a good way of kind of leveraging the expertise, experience, and also being part of a bigger global initiative to map human cells. I'm now going to focus a little bit on how we've applied and some of the challenges and also some of the areas in the next phase to particular biological context. And this is very much also hugely relevant to eczema. And I've illustrated this from the work that we've done to decode the developing human immune system, which occurs in prenatal life across many tissues and across gestational time. So this concept of space and time is very relevant to eczema as well. Patients get their lesions across multiple anatomical sites, and also the lesions evolve and the disease evolves across time. So a lot of the concepts that we've addressed in the understanding the developing human immune system is hugely relevant to how we would approach the analysis of patients with eczema. What I really want to highlight is the fact that this was very much an interdisciplinary approach with many researchers from with different expertise: immunology, hematopoiesis, developmental biology, and crucially bioinformatics. And I think in any efforts moving forwards to really make progress in eczema, we really do need interdisciplinary approaches. And that's the only way to kind of really understand and make progress at scale and with innovation. This is just to show all the manuscripts that we've published on the individual organs, including the human skin, which we published now 2 years ago. But I want to focus on a recent publication which attempted to integrate all of the information from the different organs so that we can now have a framework of the immune system as a whole, as a collective, by understanding all of that's happening across gestation time, across all of these organs. And that was a publication which came out recently. So just to sort of illustrate the different platforms that were used, and this is again all of the things that can be done for the analysis of eczema patients. And I focus first on the analysis of cells from suspension, in suspension format, i.e. from dissociating tissues, and all the things that you can measure. This is the transcriptome from single-cell RNA sequencing, and you can measure also the VDJ of T cells and B cells, including alpha beta TCR and gamma delta TCR, and also, as I mentioned, B cell receptor. So this is an integrated set of information that you can have from a particular cell and the context of the TCR and BCR for lymphocytes. Then there is also what you do with existing data sets. There are published atlases, in this case for the developing human immune system, a lot of which we've published from the different organs individually. So how do you integrate the existing data sets so that you have a better understanding of the entire framework of the developing immune system? And thirdly, additional comparator data sets. And if in the case of eczema, for example, how you might want to compare with other inflammatory disorders to have a better understanding of what's unique in eczema and what's shared with other inflammatory disorders. In this particular setting, what we did was we compared the prenatal data sets with the adult immune data sets to find out what was unique and only existed during prenatal stages compared to postnatal stages. And then there was the spatial information, and we've highlighted in situ genomics, but we also used multiplex RNA scope, which gives you in situ single cell protein level information, but more limited parameters compared to in situ genomics approaches. And finally, what's also very important is validation. And in this context, we discovered new T cell populations and B cell populations, including the human prenatal B1 cells. But in the validation, we not only used an artificial thymic organoid culture system to validate the existence of the innate T cells, innate lymphocytes that we found, but also ELISpot assays to validate the B1 cells. So all of this is going to be important in the study of any eczema patient samples. Finally, I would say that data generation is a lot easier now with more robust and commercially available platform. And one of the biggest challenges is actually in computation in terms of making sense of the data. It isn't just extracting biological insights, but how do you handle large data sets? It's a sort of a form of engineering challenge in that setting. So for this particular manuscript and study, we used SCVI to integrate all of the data sets. This is one of the newer tools to integrate data sets, taking into account of batch effects and trying to extract what is biological differences that are meaningful from technical and also other batch-related problems. And you can now see that this data set is close to 1 million cells from 25 donors and 221 libraries. And with this, you can do some more interesting analysis. For example, how cell abundance change across gestation, and in the context of patients with eczema, how this changes longitudinally. Also, organ-specific subpopulation analysis. So we may want to look at differences across sites, for example, in the skin, and also adaptive immune repertoire analysis, taking the BDJ data sets and looking at how the different T cells recombine BDJ segments and what we can understand from this process and how advanced this occurs from the perspective of B cell development. Also to then use the existing data sets to provide reference tools that other researchers can then use to annotate and make inferences of a new data set. And using the spatial information, how we can then deconvolute the in-depth information that we get from suspension analysis in terms of what are the specific tissue microenvironments that are there within specific anatomical sites. And in this manuscript, we illustrate what are the kind of tissue environments that are unique in the spleen, in the liver, etc. And in the context of the eczema, you can see how this can be used to infer the environment that's happening in the epidermis, in the dermis, in the epidermodermal junction, and so on and so forth. Another point to highlight with all of these technologies and its application is how many cells you analyze per donor and also how many donors. And to a large extent, this technology has been very expensive, but the costs are coming down and there are other ways of making the analysis cheaper or the data generation cheaper. But here you can see the number of donors that we have per organ, etc. And with the analysis of patient samples, this really needs to scale up because the next phase of where single cell technologies and its application in clinical medicine is going to be one of a multi-scale challenge in terms of being able to integrate transcriptome using single-cell genomics approaches with human genetics, DNA sequences and the variants in individuals, how that informs gene expression, and also linking it to the clinical and population level in terms of how all of these molecular and cellular processes are informing the disease phenotype and the disease experience of individuals. I'm going to focus a little bit now on what we've done with the data, and I think this is a huge opportunity for any kind of approaches for analyzing patient samples, particularly in eczema. So what we've done is to provide all of the data objects from this entire prenatal immune development as a collection that people can download the data objects for use alongside some of the reference tools that I said to kind of analyze new data sets. So this really tells you what is there, what type of data, where it's from, and also the code that we've used for the analysis. The second element of what we've done is to then also provide this data, not just as data objects, including raw data, but to be able to browse the data using a freely accessible web portal. So here you can look at the gene expression of B cells. This is CD20, and you can see that it's expressed primarily in the B lineage. There's also all the other metadata, and you can then look at the individual populations of interest and explore this. One other additional utility of these data sets, in my opinion, and what we've tried to do is to look at the expression of genes that are implicated in diseases. For example, in this context, with the human immune data set, we've looked at the expression of genes involved in inborn errors of immunity and also childhood leukemia. So you can actually click on, for example, here, the fetal bone marrow data set. There will be a drop-down menu to search for diseases. You can imagine something similar for eczema, which we've done to a limited extent with the skin data set that we have, a drop-down menu of the genes that are implicated in this particular disease. And then you can actually visualize their expression in terms of which cells are expressing them. So those are the many things that can be done. And I'm just going to finish here the overview, having talked about, what can be done now, the available technologies, the next phase, and what we can do with the data and the data objects, to finish off by thanking my lab for the fantastic years of research together, and also all of our collaborators and the many funders through the years. And thank you very much for this opportunity to present an overview on single-cell technologies. Unfortunately, I'm not going to be able to be present due to technical reasons with the IEC meeting. But if you do have any questions, please feel free to email me or get in touch on Twitter at Hanifa Lab or at Mus Hanifa, and my email is available online. Thank you.
Thank you so much for this really brilliant lecture. Sorry that you could not be here. And I'm really excited to introduce Helen He. This is very special to me. Helen was my mentee as a medical student at Mount Sinai, and now she is a resident at Mount Sinai. And I think Helen will be, she is already a rising star, but I think she'll become a key opinion leader in atopic dermatitis and other inflammatory skin diseases. As I told you, she's currently a resident at Mount Sinai in the Department of Dermatology, and she's done her bachelor at MIT in biomedical engineering, and after that she joined the medical school at Mount Sinai, and she's a resident at Mount Sinai. She published extensively on atopic dermatitis and other inflammatory skin diseases, and I'm super excited to have her here.
Good afternoon, everyone. Thank you so much to Dr. Gutman for such a kind introduction and to the IAC for inviting me to speak here today on the contribution of fibroblasts to atopic dermatitis. I have no conflicts of interest to disclose. So as this audience is already well aware, atopic dermatitis has a complex pathogenesis that involves immune dysregulation of the Th2 axis primarily, but also Th22, as well as varying degrees of Th1 and Th17, as well as abnormalities of the barrier, including epidermal hyperplasia, terminal differentiation defects, defects in lipid biosynthesis and metabolism, all contributing to transepidermal water loss. And these processes involve an interplay of various cutaneous cell populations, I think the most recognized of which include keratinocytes, T cells, dendritic cells, but fibroblasts also secrete important factors and chemokines that influence the microenvironment of the skin, but their contribution to AD has been largely not well understood. But we now know that fibroblasts also make really important pro-inflammatory cytokines and chemokines. Some of the examples are shown here. I'm not sure if you can see. I might need to use this. 1 second. Sorry, 1 moment. Oh. Sorry, thank you.
I think you can activate the laser pointer.
Is
here.
it pressing this?
And press that one. Yeah.
Let me
And then,
just.
no, then you don't need it. Then you basically.
Oh, you can see. Okay. Sorry about that. Thank you. Yeah, so some of the examples of important chemokines are shown here. For example, CCL17, which fibroblasts produce, and it's also skewing a chemokine that skews Th2, and also has been shown to be really nicely correlated with AD disease severity. Fibroblasts also make CCL11, also known as eotaxin 1, which is a potent chemoattractant for eosinophils, and CCL26, which also promotes Th2 responses and has been shown to be perhaps one of the best biomarkers of therapeutic response due to dupilumab. Other markers include periostin and CCL19, which I will discuss later, as well as CXCL12, which via interactions with CXCR4 receptor activates natural killer cells and natural killer T cells. But first, I want to discuss a couple of in vitro studies that have furthered our understanding of fibroblasts in AD. So this is a study from 2013 out of Keele University. And basically, the authors cultured together fibroblasts and keratinocytes from AD patients and healthy controls in different permutations. So they found that when they cultured together atopic fibroblasts with healthy keratinocytes, this led to increased total epidermal thickness as well as specifically stratum corneum thickness of the previously healthy keratinocytes, along with an increased expression of keratin 5, decreased keratin 10, and a downregulation of filaggrin as well. And conversely, when they cultured together healthy fibroblasts with AD keratinocytes, this partially reversed these abnormalities in the AD keratinocytes by upregulating keratin 10, decreasing keratin 5, and inducing the production of filaggrin and loricrin. So it seems that fibroblasts in AD have a role in promoting the abnormal proliferation characteristic of AD, as well as the terminal differentiation defects contributing to these characteristic barrier abnormalities, while conversely, healthy fibroblasts are able to partially rescue normal keratinocyte differentiation. And in this other study out of the University of British Columbia, the authors generated skin equivalents from healthy keratinocytes and fibroblasts that were taken from either healthy subjects or AD patients, and they found that CD4 positive T cells migrated into the dermis only of these AD skin equivalents, but not of the healthy controls. And in addition, there was significantly increased protein expression of the Th2 cytokine IL-13, as well as of the innate immunity markers IL-6 and TNF-alpha in the AD fibroblast skin equivalents compared to the healthy controls. So part of the reason that our knowledge of fibroblasts has been more limited until recently in atopic dermatitis is that the bulk of the knowledge from AD does come from bulk studies, whole biopsy studies, whereas the advent of single-cell technology has allowed us to study the gene expression profile of single cells or of specific cutaneous cell populations. So I'm highlighting a couple of publications here that utilize single-cell technology to study the gene expression profile of healthy human fibroblast cells. And each of these studies were able to identify both major and minor populations of fibroblast cells that are attributed perhaps to different anatomic locations in the skin or different functions such as wound healing, inflammation, extracellular matrix homeostasis, among others. And while there was a good amount of overlap among these different fibroblast populations, there were also marked differences as well. And the authors attributed this in part due to differences in the experimental conditions or sample processing, but also in part due to just the different characteristics of the individuals that the biopsies were taken from, such as age, gender, and especially the biopsy site, all to show that even in healthy controls, fibroblasts represent a highly heterogeneous population. And then this group out of Spain performed single-cell RNA sequencing, or they performed a reanalysis of single-cell RNA-seq data sets of healthy human fibroblast cells that were previously published. So they reanalyzed 4 data sets in total, including around 600 or so fibroblast cells that were taken from our group, from our study. And overall, in their analysis, they were able to divide the fibroblast cells into 3 overarching groups that broadly corresponded to functions in extracellular matrix homeostasis, in immune surveillance, and the group C had a more subspecialized fibroblast functions. And then they performed a second level analysis to further divide these fibroblast cells into 10 more specific subpopulations, which are shown here along with their canonical markers. And overall, while a lot of these subpopulations that they identify were only found in a subset of the original single-cell RNA-seq data sets that this reanalysis would derive from, again suggesting that either donor characteristics or sample processing conditions contribute to transcriptomic or cellular compositional shifts in the fibroblast cells that lend to the heterogeneity, and that makes it just quite difficult to classify the fibroblast cells into neat categories with consistent nomenclature, even in healthy skin. Again, just showing how heterogeneous this population is. And now we were kind of curious about how does the disease state of AD further alter these fibroblast cells. So we performed single-cell RNA sequencing on the lesional and non-lesional skin of 5 patients with moderate to severe AD and of 7 healthy controls. We were able to profile just under 40,000 cells in total, and using a clustering strategy, we divided all of these cells into 10 broad cell types, as shown here in the Tisney plot in the left. And then this heat map on the right shows some of the top markers that define each of these 10 broad cell types. But we wanted to do a second higher resolution level of clustering for each of these 10 broad cell types to gain further information about how these specific cell populations interact and contribute to the AD disease phenotype. And of course, one of the populations that we performed this analysis on was the fibroblast cells. So our reanalysis of fibroblast cells divided the fibroblasts into 4 main subpopulations, including 2 major ones and 1 minor one that was positive for collagen 11A1. That was fairly evenly represented by all of the tissue types, as you can see from the pretty even distribution of pink, blue, and yellow on the Tisney plot on the right that is color-coded by tissue type. And then there was, and then in particular, this major population of fibroblasts was actually more expressed in healthy controls compared to in AD. But I more so want to divert our attention to this unique population of fibroblasts here that, as you can see from the predominance of pink, is pretty much unique to lesional AD. And this population has a unique collagen expression profile expressing collagen 6A5, 6A6, and 18A1, and also uniquely produced the chemokines CCL2 and CCL19, suggesting that these fibroblasts have an important role in promoting Th2 responses and promoting T cell activation, trafficking, and organization around secondary lymphoid organs. And then finally, this fibroblast population also had higher expression of periostin. So periostin is an important glycoprotein that was shown in vitro studies to have its production in collagen promoted by the Th2 cytokines IL-4 and IL-13. And then periostin was then in turn shown to interact with receptors on keratinocytes to promote TSLP production, and TSLP, of course, is involved in pruritus. It's involved in perpetuating Th2 responses. So this suggests that periostin may be a critical link in this positive feedback loop where fibroblasts may be a key mediator between T cells and keratinocytes to drive Th2 responses and to perpetuate this cycle of AD disease, chronicity, and amplification. We then performed immunofluorescence to validate the presence of these inflammatory fibroblasts. So we co-stained for collagen 6A5 with both CCL19 and periostin. And as you can see, particularly in this middle panel that represents lesional AD, collagen 6A5 did co-localize with both CCL19 and periostin. And the expression is primarily in the papillary dermis, close to the dermal-epidermal junction, and the staining was much more pronounced in lesional AD compared to both healthy controls and compared to non-lesional AD. And then we also performed differential gene expression analysis, just taking the set of all fibroblast cells. So here in this heat map, we show all comparisons where log of fold change is greater than 1 and FDR is less than 0.05, red is higher gene expression, blue is lower gene expression, and just highlighting some of the genes that showed significantly higher expression in lesional AD compared to in controls. Again, we have collagen 6A6, which is one of the defining collagens of that inflammatory fibroblast population. We have the zinc finger CRIB1, which was shown in animal studies to promote Th2 responses, S100A10 and A11, as well as the pro-inflammatory cytokine IL-13, and once again periostin. And just to touch upon collagen 6A6 a little bit. So collagen 6A6 was recently shown in a study out of South Korea to have its expression promoted by IL-4 and IL-13, as well as by TSLP. And then collagen 6A6 increased expression in the dermis was then shown in turn to decrease filaggrin and loricin in the epidermis. So this again shows that collagen 6A6 seems to be unique to lesional AD, was shown in our study to be significantly more highly expressed in lesional AD compared to in healthy controls, and seems to be a link between T cells, immune cells, and keratinocyte activity. So next we wanted to see what immune cells these fibroblast populations may interact with. So then we performed a second level high resolution clustering on the set of dendritic cells. And this further subdivided into 3 different populations. Based on their canonical markers, these corresponded to a large cluster of combined Langerhans cells and type 2 conventional dendritic cells, a middle cluster containing type 1 conventional dendritic cells, and finally, this inflammatory dendritic cell population that was somewhat more overexpressed in lesional AD compared to the other groups. And here from this top dot plot here, you can also see that the proportion of dendritic cells that these inflammatory these dendritic cells represented was significantly increased in lesional AD compared to healthy controls. And then looking at the expression profile of these inflammatory dendritic cells in a little bit more detail, we see that they express LAMP3 or DC-LAMP, which is a marker of mature dendritic cells, as well as the Th2 chemokines CCL13, 17, and 22, suggesting that these dendritic cells are responsible for antigen presentation to T cells to promote Th2 differentiation. And then very interestingly, they also express CCR7, which is the receptor for CCL19 that was highly expressed by our inflammatory fibroblast subpopulation. So these dendritic cells, therefore, may be the corresponding dendritic cell population to these inflammatory fibroblasts and thus may crosstalk with these fibroblasts to promote AD-related inflammation. And next, we performed clustering analysis just on the set of all T cells. And then while there were a couple of different clusters of T cells that we identified, I mostly want to draw our attention to the one here, which represents inflammatory T cells that were overexpressed in lesional AD compared to the other tissue types. And then looking into the profile of these inflammatory T cells, we see that they are the unique producers of IL-13 and IL-22, suggesting that this is the cluster that houses Th2 and Th22 cells. And also predictably from these dot plots here, we see that the proportion of CD4 positive T cells that express IL-13 and IL-22 was either significantly or approaching significance more highly expressed in lesional AD samples compared to healthy samples. So overall, we were able to identify a novel inflammatory fibroblast subpopulation that seems to be unique in lesional AD that crosstalks with dendritic cells, T cells, as well as keratinocytes to promote T cell activation, migration, organization around secondary lymphoid organs, and also to perpetuate type 2 responses and keratinocyte differentiation abnormalities. So now I want to discuss another single-cell study that further highlighted an inflammatory role of fibroblasts in AD. So this was a large single-cell RNA-seq study that included both AD patients, psoriasis patients, as well as healthy controls of over 500,000 cells. But primarily, I want to focus on their analysis of fibroblasts. So they identified one fairly large population of fibroblasts here that is referred to as FB2 that was significantly more enriched in lesional AD compared to in healthy controls. And then this population of fibroblasts had higher expression of collagen 1A1, collagen 1A2, and collagen 6A1, as well as increased levels of CCL19 in the AD FB2 fibroblasts compared to healthy controls, and similarly higher CXCL12 here, which is not shown in this slide. But overall, this suggests that, again, fibroblasts seem to have a role in mediating inflammation in AD, and this is a pretty large subset, so perhaps there may be a smaller inflammatory subset, maybe analogous to the population that we identified that can be further teased out by a higher level, a second round of higher level clustering. And finally, I do want to discuss a very interesting study here from UPenn in which they had a mouse model with an IKKB deletion that had cutaneous features of AD. So basically, these AKKB deleted mice had in the dorsal area and ventral area cutaneous features that resembled AD, and then single-cell RNA sequencing analysis was performed from lesions from these IKKB deleted mice as well as from wild-type mice, and they were able to isolate IKKB expression to PROX1 positive fibroblasts. And it seems that the fibroblasts do induce some type of transcriptomic shift, or rather IKKB deletion does induce some type of transcriptomic shift in fibroblast cells because the fibroblasts that predominate compared to the knockout mice and the wild-type mice are distinct from each other. And just of note, the top two clusters of fibroblasts that were most highly expressed in the knockout mice were positive for collagen 6A5 and COACH respectively. And of course, collagen 6A5 was one of the markers that define our inflammatory fibroblast subpopulation. They also found that PROX1 positive fibroblasts in knockout mice versus wild-type mice had increased production of CCL19, or increased expression of CCL19, as well as CXCL12 and CCL11, CCL7. CCL11 is once again a potent eosinophil chemoattractant. And then this corresponded to significantly increased expression of eosinophils in both the skin and the blood of IKKB-deleted mice. And looking at the T cells, they found that IKKB-deleted mice had increased T cell expression of the Th2 cytokines IL-5, IL-13, as well as IL-1RL1, which is a receptor for IL-33. So taken together, it seems that this specific fibroblast population is involved in homeostasis, and IKKB deletion appears to disrupt that homeostasis and drive Th2 inflammation, T cell activation, and eosinophil activity. And while these findings were in mice, it does suggest that this fibroblast population may interact with immune cells to promote inflammatory skin lesions that resemble AD. So it'll be very interesting to see if an analogous population might be identified in human AD as well, and maybe a driver of AD pathogenesis. So in summary, while in vitro studies have already shown that fibroblasts functionally induce changes in keratinocytes and in T cells to promote immune dysregulation and barrier defects, single-cell RNA sequencing has further allowed us to have a deeper understanding of the pivotal role of fibroblasts in the pathogenesis of AD. First of all, we now see that fibroblasts are highly heterogeneous, both in healthy subjects and in AD patients. And they can vary based on anatomic location in the skin, based on donor characteristics, based on fibroblast functions, among many others. And then we were able to identify in our single-cell analysis a novel inflammatory fibroblast population that seems to be unique in lesional AD, crosstalks with immune cells and keratinocytes to promote type 2 inflammation, lymphoid organization, as well as barrier abnormalities. And more recently, this mouse model of atopic dermatitis suggests that fibroblasts may also have a role in immune homeostasis, where the disruption of this homeostasis can drive skin lesions that resemble AD. So it'll be very interesting to see if analogous population of fibroblast cells may be present and contributing to disease in human AD as well. But overall, all of these studies show that fibroblasts do seem to have a critical role in regulating signaling between immune cells and keratinocytes, and may be a key driver of AD pathogenesis, such that therapeutic targeting of fibroblasts may be quite valuable in disrupting this vicious pro-inflammatory cycle that characterizes AD disease phenotype. And with that, I, of course, want to thank Dr. Gottman, my mentor, Dr. Kruger, as well, my mentor, as well as Dr. Rana Ruiz, and the various members of our lab who were instrumental to our project, and to the groups around the world that I referenced that really contributed to our knowledge and advancing our understanding of this very important topic in AD.
Thank you so much. Brilliant lecture. Any questions from the audience? Yeah.
Thank you for your talk. I am an extension. I want to analyze for that.
Oh, excellent.
And I would like to point 2 points. One is that we have observed that one of the populations, the population you state, which expresses called 685 and CCL19, we do observe that this population is located in all healthy samples. In my analysis, which is now extended to 22 datasets, just doing a spoiler for the poster.
Mm-hmm.
In all of the datasets, this is a healthy population. It's possible that this population is also located present in AD samples, but I'm not sure if why do you find this population only in AD samples, because we have observed even in your dataset that it's also located healthy samples.
Yeah, I think I've noticed in some of the analyses of healthy samples, again, just like reflecting the heterogeneity of populations, that I see like maybe some populations that express collagen 6A5 but not CCL19 or vice versa, perhaps both as well. I think at least in our data set, we actually didn't see collagen 6A5, like CCL19 a little bit, but collagen 6A5 in our data set at least, we really didn't see in healthy samples, just in lesional AD. So again, like I think like in healthy fibroblasts probably there are like various phenotypes, and perhaps like there are like transcriptional changes that may lead to like a more inflammatory phenotype in fibroblasts. But yeah, thank you for that comment. It's good to know. Yeah.
Kenji?
Kenji, can you just hang on for the mic, so because it's being recorded? No, it's coming to you.
Very beautiful talk. So I'm interested in the inflammatory fibroblast. So what do you think the origin of inflammatory fibroblast and where they are located in the skin?
Yeah, so when we did immunofluorescence, we found that these fibroblasts are primarily in the papillary dermis, kind of very close to the dermal epidermal junction. So in terms of spatial location, that's where I think it's coming from. In terms of, what their function is or what their role is, where they come from, I think, collagen 6A5, as discussed, was present in some healthy samples in other studies, but I think they may have a role in like destabilizing the heterotrimers of the collagen and maybe contributing to barrier defects in that way. Where they're coming from, a little hard to say if maybe this is a very distinct like original cell type versus a very severe transcriptomic shift in the original healthy fibroblast cells to create this unique phenotype where these unique collagens are expressed and like having this inflammatory markers. But yeah, it'd be interesting to further explore where they come from.
The reason why I'm asking is in the chronic phase of atopic dermatitis, dendritic cells localize usually around hair follicle or post-capillary venules. So I just wonder, CCL19, the source of the chemoattractant for dendritic cells, maybe so inflammatory fibroblasts might be located around blood vessels. So
yeah, at least in our staining, I don't think we noticed it around the blood vessels, but I think that's definitely a possibility given that, you say like dendritic cell populations are already localized there as well.
Sorry,
Yeah.
the last question is, you did single cell RNA sequence. So what is a major source of CCL19?
Like which cell types are expressing CCL19? I would say that primarily the fibroblasts are actually the largest source in lesional AD, and then the parasites and vascular smooth muscle cells were also making CCL19 as well.
Thank you.
Thank you.
Emma, I have a question, if I may. Beautiful talk. Thank you very much. So following on from Kenji's question, did you have chance to look to see if your inflammatory fibroblasts were co-localized and adjacent to the dendritic cells in that zone? Were they physically opposed?
So actually, I don't think we did staining for dendritic cells, but we did, not shown in my slides, unfortunately, but we did show that they co-localize really closely to CD3 positive T cells. Okay. So we were able to demonstrate like co-localization with immune cells in general, but I think staining for dendritic cells would also be really valuable.
Yeah, I think we did it in other studies, and I do think they co-localize.
And another question, if I may. So, given the heterogeneity that you demonstrated, I wondered if you'd also looked at their filaggrin status to see if that associated with any particular types of the fibroblasts.
Yeah, that's a good question, actually. I do not believe we knew the filaggrin status of the patients that we had, but I think that's a good direction in the future to compare like filaggrin mutation versus lack of filaggrin mutation.
Yeah, it was a small study, so we didn't feel it will be beneficial for this study, but I completely agree. In large data sets, definitely it should be done. Yeah.
Just answering your question,
We're going to record it, so it will help.
Okay, thank you. So in normal fibroblasts, there's one axis which has two main populations. One of them are fibroblasts which are primed to respond, and there are the second fibroblasts, normal fibroblasts, also in AD, expressing CCL19, which we believe they are like late response fibroblasts. So the first fibroblasts, the attack cells, they are related more to neutrophils and primary responses, and the CCL19 fibroblasts, they come back later, just are more related to DCs and other types of immune cells.
Yeah, I mean, that makes some sense because all of our AD patients are chronic AD. So they've had AD for basically all of their lives from ages like less than 5 years old, basically. So that's another good point for a future study to compare like patients with more acute AD that develop in adulthood versus chronic AD from childhood.
Yes, there is a question there.
Yeah, hi, this is
Hi.
Matthias Hübenthal from Kiel, Germany. Thanks for the nice talk. I was wondering, so talking about the fibroblast clusters you identified, do you have estimates for the stability of this clustering? So if you remove either samples or cells, so how stable is the observation and or the clustering?
Yeah, I mean, we actually perform like various levels of analysis and like, to clean up the cells and make sure they're all good quality. And then, we, of course, as I mentioned with the heterogeneity, age, gender, all of that really could affect the fibroblast population. So we did like various sensitivity analysis where we, only took like age-matched patients or like only, or like gender-matched patients, things of that nature. They were all taken from the same biopsy site, so that wasn't as much of a factor. But we noticed that the main clusters that we formed were pretty much identical. There may have been small like transcriptomic shifts, and that's another interesting thing to look at for single cell, like with age, like how does the transcriptomic profile further vary. But overall, the main clusters that we identified were pretty constant regardless of these covariates.
I see, and did you perform cross-validation, so classical type of, so to say in silico replication of the clustering?
Sorry, can you repeat that?
So were you able also to perform cross-validation or something like this, so that you in silico, so to say, can validate the classification?
I do not know
we also
what
can
you
do
mean
this
because
offline.
in single cell we applied similar methods to other studies, so not sure what you mean by silico validation.
We probably also can discuss this offline.
Of course, happy to.
Yeah.
Thank you.
Great, thanks.
Okay, I think we should conclude this beautiful talk. Thank you so much, Helen. So my distinct pleasure to introduce the next speaker as well, also close to my heart, Dr. Andrew G, that we recently recruited from Stanford. And very similar to Helen, actually, he also did his bachelor at MIT. So you see, there is a plus to be knowing how to analyze your samples and be a bioinformatician and a physician. So Andrew G is an assistant professor of dermatology at the Icahn School of Medicine at Mount Sinai. He did a little bit unusual path that usually dermatologists do not do. Usually they do postdocs before they go to dermatology or research before they go into dermatology. But Andrew actually was already a dermatologist. He trained at Stanford. He did medical school at Cornell, trained at Stanford, and then he did a postdoc in Paul Kavari's lab after he was a dermatologist, just showing his commitment to being a physician scientist. And his lab is focused on multi-omic approaches, spatial transcriptomics, and single cell, 2 publications in Cell recently. He certainly is becoming one of the key opinion leaders in spatial transcriptomics, and we are very fortunate to have him in my department and here with us.
Thank you so much, Dr. Gutman and Dr. Reynolds, for the invitation to speak at the symposium. I joined Mount Sinai about a little over a year ago now. Started a lab there and still see patients 1 day per week. And so it's exciting to be part of an exciting department going forward. no disclosures. So I actually wanted to start the talk by taking a step back and thinking about the big picture a little bit. And the application of all these multi-omics technologies, really one ultimate goal, I think, of these tools is to achieve personalized or precision medicine, or precision dermatology, I should put it. And really what that is being able to match a patient with the right therapy. In the broadest sense, I think this is automatically going. But in the broadest sense, simply making a diagnosis of atopic dermatitis versus psoriasis, let's say, and saying we should put this patient on dupilumab as opposed to secukinumab, is sort of the broadest level of that. But I think we're all here because we think we can do better than that in trying to understand intra-disease heterogeneity. Why is it that some patients with atopic dermatitis respond or don't respond to dupilumab? And so thinking about how to get there, this is where I think multi-omics technologies can help us. So this is just an illustration of all the different levels of integration, types of data you can integrate to achieve a better understanding of stratifying patients and being able to match them to specific biomarkers or response rates to different therapies. And I think this is one example, one analogy, one visualization of this. And I know you've seen a lot of analogies today, but I like to invoke this one from Howard Chang, who kind of likened this to Google Maps, in that all of the different biological assays you can perform on tissue can be integrated to get a clearer picture, a deeper picture of any particular, in this case he called it a personal regulome, of how cells are interacting and behaving within a tissue. Just like in Google Maps, you do have to take many different data surveys, ultimately integrate all those together to actually get a clearer picture of what's happening in the real world. So I think one of the most powerful biological insights and goals of being able to use these technologies is to piece together intercellular communication. And this is really something that's been well established as a success story across inflammatory skin diseases, well-known situations like psoriasis and atopic dermatitis, where when you block the ability of one cell to communicate with another cell through blockade of some of these major cytokines driving the disease, we can see that they translate into therapeutic success. And so one of the major steps towards understanding intercellular communication, as has been mentioned in the symposium, is the advent of single-cell RNA sequencing. And so this is a running tally that I've put up over the past 4 years, not a comprehensive list by any means, but some of the papers that have been published just on normal or diseased human skin, tally about 11, 12 publications here. And it's easy for me to imagine that, even a few years from now, that you could put up a similar slide showing all the publications that might be coming from spatial transcriptomics, which I think is a logical next wave. And one of the inherent limitations of single-cell RNA sequencing is, of course, when you dissociate the tissue into single cells, you inherently lose their spatial context. And that's where something like spatial transcriptomics can really come in and piece together where these cells came from to localize them. I'm showing you one workflow we've established in our lab to try to get at this question of studying cellular communication using bioinformatics approaches to integrate single-cell and spatial transcriptomics data, and then doing something like CRISPR screens to perturbationally try to disrupt the communication and see the functional effects of the communication. And I won't be talking about that mostly today. I'm going to focus on spatial transcriptomics, which Dr. Reynolds mentioned was the method of the year in 2020. Surely this audience has heard of this technology, but I'll give a brief overview of the different types of spatial transcriptomics technologies that exist. In this case, I'm showing here the 10x Visium platform, at this point, probably the most widely known or widely used technology, where you have these capture spots arrayed on this slide, which each have basically a poly-T primer that can capture the mRNA. mRNA is permeabilized from the tissue, falls down, binds to these spots. Each of these spots has a unique barcode that when you generate the cDNA and amplify it and prepare a library for sequencing, you then use that barcode to positionally map back the transcripts to their position in space. And so I do want to emphasize that actually Visium is just one of several different spatial transcriptomics techniques. And so I'll just put a shameless plug for an article our lab worked on for JID, a Research Techniques Made Simple article that overviews different types of spatial transcriptomics. And then as a postdoc, I worked on a review that overviewed the computational algorithms and techniques for integrating single cell and spatial data together and trying to maximize biological insights. So as I mentioned, spatial transcriptomics does cover a suite of techniques, and they kind of fall into three major categories, which Dr. Hanifa also mentioned in her talk. But in situ hybridization is shown on the left here. This is using probes that target known transcripts ahead of time. You can design these probes to target several hundred, at this point, up to several thousand pre-designed probes that will target transcripts of interest in a tissue. In situ sequencing is probably maybe the least well-known at this point, but the 10x Xenium platform that's coming out, I believe later this year, is based on this technology, which basically uses hybridized probes that are barcoded uniquely so that you perform in situ sequencing of the probes with these fluorescent tagged oligos. And through the assignment of the barcodes to transcripts, you can then deconvolve all the different transcripts that are in your tissue. And then 10x Visium is a prominent example of an in situ capture method, which relies on the poly-T unbiased capture of RNA transcripts that have fallen onto the specialized slide. So what's important to remember about these techniques is that they each have their own advantages and disadvantages, trade-offs and limitations. And so briefly, the things you want to think about when you're picking one of these to perform on your tissue, first question would be resolution. And resolution basically refers to how accurate or how exact of a location you can assign that transcript. And so prominently, 10x Visium has a 55 micron diameter spot size, meaning its resolution is 55 microns. You cannot tell within that 55 microns where exactly that transcript came from. 55 microns is not, it's not huge, but you still capture up to tens of cells in there. And so it's not possible to say which of those tens of cells this transcript came from. Conversely, with targeted approaches like MERFISH, which is probably the most prominent example of in situ hybridization, StarMap, which I think is, I'm not sure, but I think 10x Xenium is based on this technology. These using probes can actually achieve subcellular resolution. The probes are about 10 nanometer resolved distances. So you can really say, where within a particular single cell this transcript came from. And then the other things you want to consider are breadth or depth. So by breadth, I mean, do you want to capture transcriptome wide in an unbiased manner like 10x Visium? Or do you really want to go for well-known targets but probe them deeply? And actually, this figure on the left is a prime example of the breadth or depth, quandary. This is a piece of mouse brain tissue that is probed with the in situ hybridization technique. The same gene is shown with the 10x Visium technique, and you can see that this is clearly a deeper capture of all the RNA transcripts that are present, whereas 10x Visium really just samples in a fairly shallow manner, not as many of the transcripts. So that's what I'm referring to for breadth or depth. Of course, the disadvantage of something like an in situ hybridization technique is that you're limited to the number of targets you can capture as opposed to this unbiased transcriptome-wide. And then another consideration is field of view. That just means how large of an area of tissue you're profiling, whether you're focused in on a very small area of the tissue or capturing tissue-wide. I will say that nowadays all of these techniques do a pretty good job at getting tissue-wide capture, so this might be a little obsolete of a question. And then finally, this may be the key here to unlocking the clinical power of these techniques, is whether they can be applied towards fresh frozen tissue or FFPE archived tissue. And so all of these techniques currently do better in fresh frozen, but they are all being developed for FFPE, and it'll be interesting to see which one wins this sort of battle. And so, in my opinion, right now, as of today, in terms of the clinical applications of ST, they really are stronger as a research tool. But as a research tool applied to human tissue, is about the best we can get in terms of a deeper look into what's going on within human tissue. And so I'll highlight over the next few minutes a couple use case scenarios with real data that we've generated to show you sort of 3 different goals or applications of this as a research tool in terms of better annotation of cell types, identifying niches or neighbors of cell types that exist in sort of a non-random pattern, and then, of course, intercellular communication. And so as I speculate a little bit about the clinical applications of ST, I think they'll probably prove their worth as biomarker tools in well-designed clinical trials, either prospectively or some of these retrospective, prospective design trials taking archived tissue. I'm not quite sure about a direct clinical assay, which is, I'm sure people are thinking about it. I think there are a lot of steps that need to be taken, a lot of hurdles to clear before it becomes more of a direct clinical assay. So here's some data from 10x Visium on normal skin that we generated. And what I want to highlight here is that the transcriptomes are represented as spots that overlie a tissue. And because each of these spots has a transcriptome associated with, you can treat it very similarly to single-cell RNA sequencing data and cluster these spots. And what happens when you do that is the spots actually end up recapitulating the histologic architecture of the tissue. So we see, for example, there are these 2 clusters that really nicely overlap the interfollicular epidermis, the superficial layer of the skin. Then you have these other clusters that overlap with the hair follicles that are prominently visible in the histology of the section. Likewise, adnexal structures like sebaceous glands and eccrine glands are also captured transcriptomically within the tissue. So you can see that the transcriptome of the tissue corresponds nicely to the histology. But as I mentioned before, you really do get synergistic power in integrating these data with single-cell RNA sequencing data. So to kind of get at that, we actually integrated 4 different published data sets of normal human skin and kind of came up with a more consensus clustering of cell types from human skin across 80,000 cells and 24 donors. And when we integrated this data with our spatial transcriptomics data, basically projecting and scoring these clusters onto the tissue, we get some interesting insights. A lot of expected things happened. For example, different clusters associated with the IFE that I showed you before projected nicely to the IFE. Likewise, there are some single-cell clusters that projected nicely to the hair follicle regions, including outer bulge or inner bulge of the hair follicle, as well as more of the upper hair follicle, more like infundibulum. Sorry, this is going on its own, like a runaway train. But interestingly, what we found was that there were these 2 clusters that we didn't really know much about based on their gene expression analysis. But when we used spatial transcriptomics, we found that they really projected nicely over the eccrine gland. And just to illustrate that a little bit further, what we saw is that there were these areas in the histology that looked that were clearly visible for eccrine glands. Again, I apologize that this is just going on its own. We scored those 2 clusters and found that they scored almost perfectly aligned with the histological areas. And what we found was that this particular cluster 16 was previously annotated by Jeffrey Cheng's group at UCSF as an IFE channel cell, which actually now we can infer based on our analysis are actually eccrine duct cells that are traversing through the IFE. Of course, that's where eccrine glands empty. And this was confirmed by expression of keratin 77, which was uniquely in this cluster. When we looked in Human Protein Atlas, keratin 77 was uniquely expressed in eccrine ducts and not secretory cells. So this is just an example of how spatial transcriptomics data lends additional insight into something you might not be able to infer from just transcriptomics or gene expression alone. And so I'll turn to some of our data in squamous cell carcinoma, where we basically profiled 10 patient tumors in their matched normal skin and deeper analyzed the keratinocyte subpopulations. And what we found was that normal and tumor keratinocytes both contain subpopulations that resemble each other, including cells expressing basal markers or cycling markers or differentiating markers. But in tumors, we found this unique expression signature of a certain subpopulation of cells expressing genes that were basically minimally or not expressed at all in normal skin. And we called this particular subpopulation of cells tumor-specific keratinocytes because of that unique gene expression signature. And so here's a heat map that just demonstrates that these particular cells expressed, upregulated these genes that were really minimally expressed across other tumor subpopulations as well as normal skin. And we inferred that these were likely to be an invasive subpopulation based on their expression of some very classic epithelial mesenchymal transition or EMT genes. And when we scored these cells for an EMT signature of 200 genes, we found that they did score the highest across any other subpopulation. We wanted to know whether this was some quirk of squamous cell carcinoma of the skin or if this had anything more broad implications across other cancers. So we looked in the TCGA, the Cancer Genome Atlas, and we scored various epithelial cancer types for our TSK signature, and we found that across 6 of these different types of cancer, including head and neck squamous cell carcinoma, kidney cancers, mesothelioma, and pancreatic adenocarcinoma, that a higher TSK score was associated with worse overall survival, suggesting that this was a broader phenomenon across other cancers. And interestingly enough, we found a cohort of patients with head and neck SCC and lung SCC treated with anti-PD-1 therapy. When we scored those patients for TSKs, we found that those that scored more highly were associated with resistance. So we became really interested in the subpopulation. But the first question we wanted to know was, where was this subpopulation located? We had done some inferring that they were invasive. And so in spatial transcriptomics, what we found was that this population was present at the leading edge of tumors in a very specific and enriched at the leading edge. And we did this through integrating the single cell RNA sequencing data, where we scored our spatial transcriptomics spots for our single cell RNA sequencing patient signature, and found that there was this particular cluster of spots that was highest scoring for TSKs, and that corresponded nicely to the tissue architecture, as well as this unique TSK-specific gene, MMP10. And then we asked what other cell types were potentially surrounding these TSKs. And for that, we also turned to our spatial transcriptomics data. We found a different cluster of spots that exhibited nearest neighbor localization to these TSK high spots. And when we scored these spots for our different cell types in our single cell data, we found that these particular spots were enriched for CAF and endothelial-like genes, CAFs being cancer-associated fibroblasts. So we think we found this TSK CAF endothelial cell niche, and we wanted to know potentially how those cells were communicating with one another. So very classically performing ligand receptor analysis, but layering in the spatial data as well. So what that looked like was looking within the single cell data to look for a specific expression of ligands and corresponding receptors. So in this case, I'm showing you the expression of SPP1 in macrophages in the tumor versus its receptor, integrin beta-1, its expression in tumor cells. And in the spatial transcriptomics data, we can see that a lot of these ligands and receptors are co-localized in the same regions of the tumor. And so imagine doing this across thousands of ligands and receptor pairs. You can start to put together these communication maps that outline different ligands being expressed by different cell types within your samples, their matched receptors, and then whether they exhibit spatial localization with one another. And so here just highlighting that SPP1, which I showed you previously, were actually expressed by both macrophages and MDSCs, and its receptor integrin beta-1 was upregulated in TSKs. And in this particular heat map, the purple represents whether it's significant in single cell analyses, and the star indicates whether they exhibit spatial localization. So you can see how you can start to piece together all these different ways all these various cell types might be communicating with one another to get a better sense of intercellular communication. So then what's next in terms of these technologies? Well, the resolution question is being addressed by the advent of, or the development of, smaller capture spots down to 2 microns, which 10x is working on at the moment. I think they're releasing it sometime in the spring. 2 micron resolution does achieve effectively subcellular resolution. So this idea that the 55 micron spots aren't highly resolved enough, this is their way to address that. And then even smaller, our recent Cell paper described this technology that achieved up to, I believe it was 200 nanometer spot sizes. So that's all happening. In parallel with that, the advent or the development of computational algorithms to effectively be able to handle these new technologies really represent a coevolution of both the chemistry on the left and the bioinformatics on the right. And so this was a paper that actually already has done some benchmarking of different ways to integrate these data, single cell and spatial data together. As an example of single cell resolution ST, we recently teamed with VizGen, makers of MERScope, a commercialized version of MERFISH, to generate some data from human skin. And here is some of that data shown on the left here, the expression of 6 different genes. And then on the right, what you can do with these imaging data is segment the image into single cells using machine learning algorithms. So basically, this machine learning algorithm will actually take this image, break it up into single cells. And because these are associated with up to hundreds of genes or so, you can also treat them just like single cell RNA sequencing data and cluster them and perform UMAP dimensionality reduction, clustering to help annotate these cell types, and that's what we've done here. Where on the right, we really clustered and generated UMAP and then projected their identities back onto the tissue itself. And so that's all I wanted to, I got spoiler alert, I was gonna thank my daughter who was born August 15th. But you can see she's a newest contributing member of the team here. She's, astutely giving me feedback on some of the data that we've generated. But this is work that we started at Stanford and carrying on at Mount Sinai, and appreciate all your attention. Thank you.
Amazing presentation, really, and it's amazing that he came in person. Indeed, he has a month-old baby at home, so really, we are very pleased that you are here with us. Any questions from the audience? Yeah.
I'm Monica from Jensen.
Hi.
Hi, would you just mind waiting for the mic?
I can hear you, but I don't think the mic's on.
Help me repeat the question because your mic works.
We hear you, I think.
Yeah.
Just speak loud.
I can repeat it. I'll repeat.
So my question is, the FISGen is, very exciting, more single cell-like, resolution. With, let's say, 500 genes, do you think we can kind of reproduce those clusters that we see by dissociated skin tissue single cell RNA sequence?
Yeah, so the question was, with a targeted panel of 500 genes or so, can you recreate the unbiased nature of single-cell RNA sequencing data? I think the answer to that question is actually yes, but obviously we need to show that with the analyses. The common algorithm, the common workflow for single-cell RNA sequencing is to identify the top 2,000 or so variable genes before you carry that forward. So you're not really using the entire transcriptome necessarily for a lot of those analyses, although, I'd love to see more formal proof of that, that the whole transcriptome doesn't matter. But I think 500 genes is sufficient to deconvolve all major cell types, but the key is you have to design that panel yourself. And so the burden, the challenge with these targeted panels is the burden now falls onto the investigator what genes to include. And of course, you might miss some that might be important. I think that, all of these techniques, it's probably going to be helpful to generate single-cell RNA sequencing data along with it, just because it's so well established. It works really well and provides an additional orthogonal validation. So, nothing is perfect. I think that 500 genes, though, can get you a long way.
Yeah, I completely agree, and Andrew and I often have these discussions, and sometimes you may need more than a panel of 500, depending on what disease you are looking for. So you need to keep that in mind, because you have a little bit differences in cell types. Yeah.
So thank you for, I really enjoyed your print and reading it, and it was an amazing work.
Thank you.
My question is, do you see like a practical application of spatial transcriptomics in the clinic, like from 5-10 years now on? Would you give me an example of someone that arrives at the clinic and a spatial transcriptomic analysis is applying onto them to do a diagnosis, or what do you do?
Yeah, so the question is, projecting 5 to 10 years from now, can you think of a clinical application of a spatial transcriptomic assay? It's a great question. I've thought a little bit about this. I think there are a lot of factors to consider, and so one question is how many genes do you actually need to assay to actually gain useful information? That's probably the biggest question that will determine, which of these techniques makes it in, which are most practical. I think there are some advantages to RNA over protein, for example, I think, just because with protein, if it's secreted, you don't quite get cell type localization, versus RNA probing, you should be able to get the exact cell type that's expressing the gene of interest. Again, how many genes, I think, will be the key. The other barriers are just standardization across tissue types. We're working with skin now with some of our MERFISH efforts, and it's actually quite hard to prepare these samples and get high quality data from them. It's a big challenge in terms of how to prepare the samples. It takes, a lot of very bespoke steps, and so that will probably vary across different tissues. I think skin, I think, is actually the toughest type of tissue to work with, to be honest. So yeah, it's hard to say. I think, don't want to reveal all my ideas in a public forum, but I don't know. It's a good question.
Can I follow up with a kind of slightly related question, which goes back to your first slide, which was to get to personalized medicine for individuals. And so we know from studies done in inflammatory skin disease that you need at least 50 subjects per condition to start to be able to tease out biomarkers that are reproducible and can be validated. So in that sense, I wonder whether the integration of the data could also go backwards towards bulk to enable you then to get greater insights. So you've been projecting onto the spatial, but I wondered if you could also do the reverse to enable you to get sufficient sample numbers that you can then start to figure out your subgroups, which is going to be essential for personalizing your therapies.
Yeah, that's a great point. I think there are not a lot of formal power analyses in determining, the single cell RNA sequencing studies. People just guess how many patients you need to really see an effect. And so I totally agree with you. I think, right now these techniques really lend themselves to smaller cohorts, going deeper, but really to unlock the precision medicine aspect of it, you got to get larger cohorts. So whether that's, a lot of independent groups doing their own studies and then integrating all those patients together, that could be one way forward. But yeah, I think it's unclear at the moment. I think large consortiums like the Human Cell Atlas are essentially trying to achieve something along those lines. Maybe we need something like that for diseased tissue as well.
Completely agree. Okay, so I think if there are no more questions, maybe we'll take a break and we'll come back in 10 minutes or 15 minutes.
Yeah, let's make it 15 minutes if that's okay, because people may also join us who are expecting a start around 3.
Yeah.
So, and it's always nice to have a bit of a break
Definitely.
and refresh.
Yeah, very exciting first part.
Yeah, thanks for your interesting questions, and we hope to keep it interactive going forward. I wonder if we could please reconvene so that we can get going with the second session. So thank you for making the first session so successful. Great participation from the audience. And I think it's been a really great session so far. So we're going into part 2. And it's a great pleasure for me to introduce Stephanie Eirich, who's a non-clinical biologist by training. And she's an expert in communication between immune and epithelial cells, which you've already heard quite a lot about. And she's been applying these new technologies to try and understand in greater depth inflammatory skin disease. And as we've heard as well, she's then aims to translate the results of these findings into potential diagnostic tools as well as therapeutics. So we welcome you to the podium.
Yeah, thanks, Nick, for these words. And you already heard a lot today about spatial transcriptomics. And please forgive me that some of the things that I will tell you, you will have heard before. I was asked to answer more or less the questions if spatial transcriptomics can be applied also to atopic dermatitis. And what did I do now? It's this one. And as we already heard, spatial transcriptomics is quite a new technology. And I was just going to PubMed and searching here, and especially in skin research, it seems to be really a new technology. Just 61 hits from 2012 to now. And as we also heard, it's the method of the year 2020. And I guess this gives you an implication that we should start using this technology in skin research. Also, this is a kind of picture of the fruit salad that we heard already from Moose in Lego bricks shown here. So bulk RNA sequencing is a fantastic technique that has actually revolutionized our understanding of disease pathology. However, the mRNAs that we picked there, we do not know where they come from. So the single cell technology actually helped us a lot to understand our mRNA signals that we retrieved from bulk sequencing. And spatial transcriptomics actually brings all these bricks together so we can understand how the cells are residing, or where the mRNAs are residing in the tissue, and how they might potentially communicate with other cells. The technology, you also heard about that. We were working with 10x, the Visium technology, so that's why I'm showing it here. You can apply to fresh frozen and FFPE tissues, and it's quite a straightforward workflow. So I guess you could implement it in your research lab as well. So you have these capture areas. In every capture area where you put your sample on, you have 5,000 spots where you find these barcodes. We already heard that these spots have a diameter of 55 micrometers, and we already heard from Andrew that this is actually one of the big disadvantages of the technology so far. So then you can have your barcoded mRNAs and analyze the sample. We already saw pictures like that. So that's how we analyzed chronic inflammatory skin diseases. And here you see all these spots that actually cover the whole tissue. So what did we do? We were interested in chronic inflammatory skin diseases and built up a cohort of spatial transcriptomics for lichen planus, for atopic dermatitis and psoriasis. And we manually annotated our samples as well, but also used, of course, the automatic annotation that the analysis tool actually provides. And in total, we had 90 samples and more than 60,000 transcriptomes to analyze. As you heard before, the interest of our lab is mostly in understanding the communication of immune cells with epithelial cells in the tissue, and naturally we're interested in cytokines. So we were, first of all, looking at the expression of cytokines in our inflamed skin. And you see actually not many spots. And we heard it from Andrew before that this might be also due to the resolution and the resolution and also the capturing of the technology. But nevertheless, we were quite intrigued to see these spots covering or being dispersed over the tissue. And what you nicely can see is actually that interferon gamma here is mostly attributed to lichen planus, what we would expect from the pathology knowledge that we already have. IL-13 we find in atopic dermatitis, however, no transcripts for IL-4, and IL-17 is nicely expressed in psoriasis. However, when we look to the counts of each single mRNA in a section, the counts are not really high. So maximum 37 counts for interferon gamma, which is just a handful of counts for a cytokine transcript, yeah? And also IL-17, you might remember stainings of IL-17 in psoriasis skin. The skin is really full of this protein, but however, the cytokine transcripts are quite rare. When you have a look onto the cytokine transcripts of these three cytokines and compare it to all the transcripts of other genes we find in a lesion, then you see that there is a quite high fold change difference. And in total, the cytokines are 125 times less expressed than all the other genes. So we were surprised by that and then annotated our tissue. We did it manually. So you see here nicely the basal epidermis in orange, then the middle epidermis and the upper epidermis. And you can also annotate the dermis depth. And by this, we had the possibility to actually look into the expression. So where are the cytokines expressed in the tissue? You see here, you might wonder what these big dots are. That is a housekeeping gene, a reference gene. In our case, it was GAPTH to just show how much more these reference genes are expressed. Lesional, non-lesional skin, we would expect that. There is not much going on concerning cytokine expression. However, in lesional skin, we clearly saw an upregulation of cytokine transcripts. And interestingly, we found interferon gamma mostly at the junction site of epidermis and dermis. The IL-13 is more interspersed between epidermis and dermis, and IL-17 is clearly dominating the epidermis. But again, the transcripts we found were quite low. So when we just focus here on lesional skin, we found 300 transcripts of interferon gamma over all our cohort that we investigated, and less than 100 for IL-13 and IL-17A. So of course we were wondering if this is actually just an artifact that we're looking at, or if this is a meaningful expression that we found. So we went to our single-cell data sets that we also had in our lab, and we actually could confirm the findings that we had in the spatial quite well in the single cell, because also here we found quite low numbers of transcripts for interferon gamma, IL-13, and IL-17. And as we heard already before, we also used another spatial technique, which is in situ hybridization. And also here we could quite confirm the results that we saw in the spatial transcript complex, because also here we did not find many transcripts for interferon gamma, IL-13, or IL-17A. But interestingly, the transcripts were distributed disease-specifically, meaning that the lichen planus actually expressed most of the interferon gamma transcripts, and most of the IL-17 transcripts were expressed in psoriasis. So this was expected. We also heard about this issue that a spot has a diameter of 55 micrometer, and of course that naturally contains more than just one cell. So we were using the Tangram algorithm to understand the cellular composition now of our spots, and to also understand which cells might now produce these cytokines. And we already heard it. That's the data set that comes from Moose Hanifa's lab that we used. It's a single-cell data set that we used as input for our deconvolution of our cytokine transcript positive spots. And this is just shown here for IL-17A. So that are all our spots that express a cytokine IL-17A transcripts. And when we look into these spots, we see the cellular composition. It's just a prediction to remind everybody of you. So interestingly, in the epidermis, most of the cytokine transcripts are expressed in spots that contain antigen-presenting cells and lymphoid cells. And mostly the spots in the dermis contain lymphoid and mast cells. And when we draw a bit more, a view more closely on the lymphoid and mast cells, then you see here the orange spots that are actually all those spots that contained T cells, followed by some innate lymphoid cells and NK cells. So by that, we could be quite sure that actually cytokine production in the tissue goes along with T cells that are also present in these spots. Then we were wondering if it's really possible now with spatial transcriptomics to attribute a specific activity or function of these low numbers of cytokines in the tissue. And for this, we were using actually an ancient data set that we already had available in the lab. We used 2D keratinocytes that were stimulated with recombinant cytokines. And we performed gene expression arrays that are already outdated nowadays, I would say, and identified specific signatures, so differentially expressed genes for IL-17, interferon gamma, and IL-13. And then we used these differentially expressed genes from a 2D in vitro culture, and we're checking if we find these genes also in our spatial spots that express cytokine transcripts. And the merge of these DEGs we use then as specific cytokine signatures, so interferon gamma, IL-17 and IL-13 response genes. And that is just the overview of the genes that we have been using, and some of the genes might be familiar to you. So for IL-17, we included DEFB4, S100 proteins, IL-36, for instance, CXL9, 10, 11 for interferon gamma, and just 4 genes here for IL-13. So that was actually our weakest response signature that we could obtain. And so we thought, to make it a bit more stronger, it makes sense maybe to add some genes from the literature that are known to be associated with IL-13 function. So now we have these specific response signatures for each cytokine and thought, now let's correlate the cytokine transcripts with these responder gene signatures. And this is what we did over all the spots that we had available. And it was quite nice because we could see a kind of correlation here for interferon gamma transcripts and the responder genes. But actually, the correlation was not really overwhelming, I have to say. Oh, no. How can I go back? Now I'm there. Especially for IL-17, it was really, really weak. And then we thought, why not use the beauty of spatial transcriptomics and look into the tissue where actually the cytokines are expressed? And this is what we did. We developed a spatial density clustering method. So first of all, we marked all the spots that express the cytokine. And if spots are neighbored, they were put together as a cluster. And you see here the clusters of radius 0. And then we put around different radiuses. So we added in this radius 1 here the next neighboring spot and analyzed the responder gene signature. Here we added the next 2 spots and the next 3 spots and so on. And by this we could actually understand that cytokines do have a different radius of action in the tissue. So when we just focus here on interferon gamma, this orange line, you see that interferon gamma seems to have not a really specific radius of action because, first of all, we see a lot of expression in the tissue, and we all know that interferon gamma per se is quite an active cytokine. IL-13, on the other hand, had a peak radius of 3, so the perfect action of IL-13 was in the radius of 3, and then the activity declined. And most interesting was that IL-17 here, the response signature for IL-17 genes actually peaked at radius 0, so where the cytokine was present. So IL-17 really had a strong or a small radius of action in the tissue. And then we used this density clustering method to analyze again the correlation. So we used the optimal radius for each cytokine and correlated those radiuses with the responder genes that we identified. And you see that we could boost the correlations for each cytokine, especially for the IL-17, and we could also boost the significance of this correlation. And this means, in essence, that we find low numbers of cytokine transcripts. So far, we do not know why exactly. Is it the technology or is it real, what we find there? But what we can say is that we can attribute these low numbers to a function, and we see that these low numbers of cytokines actually can boost and amplify inflammatory responses in the tissue. Then we were intrigued if we can use spatial actually to identify new biomarkers or potentially new therapeutic targets. And I would like to show you just some recent results where we tried this in atopic dermatitis. So what did we do here? We used our IL-13 positive spots. Now we knew that the perfect radius of action of IL-13 is radius 3. So we analyzed then all DAGs that were actually compared between the radius of 3 and all other spots that we had in the tissue and identified potentially new responder genes or genes that are related to IL-13 function. And in total, we identified 148 genes, and these here are depicted that are just the ones that had the highest fold change and the highest p-value. And I just want to draw your attention to these twos, the CLAC10A, acetyl-lactin domain family member, and FPR3, which is a formal peptide receptor. And I would like to show you how this looks in the tissue. So this is an atopic eczema slide with just 2 dots of IL-13 transcripts. And when we look at the CLAC10A expression in the tissue, you nicely can see that the CLAC10A expression actually peaks here around these IL-13 positive spots. And you can draw a nice cluster around that. CLAC10A is maybe known for some because it is involved in type 2 differentiation of macrophages, and by this is already a bit attributed to this type 2 kind of inflammation. However, for this fermial peptide receptor, there is no association known with atopic dermatitis so far, and also here you nicely can see that expression of FPR actually peaks around these 2 IL-13 positive cytokine spots. So let me come to my take-home messages. I hope that I could show you that lesional skin harbors low numbers of cytokine transcripts. And these cytokines have a specific radius of action in tissues. This might be different in skin and other organs, that's for sure, but in skin we could identify these perfect radiuses. And that a few transcripts are actually able to induce thousands of responder transcripts in their close proximity, and by this amplify inflammatory responses in the tissue. Then I thought maybe it's worth to report a bit about the pros and cons of spatial transcriptomics, and we already heard that also before in the talks, but I guess it's a really beautiful technique that allows high throughput tissue profiling. We can analyze now transcriptomes in their native context, and we can understand communication pathways in a depth that we were not able before. It is also already possible to combine spatial transcriptomics with protein analysis, so you can already stain your tissue with up to 6 protein markers, and by this also identify your cells in more depth than just doing transcriptomics and the Tangram algorithm, but also there is, of course, room for improvement. We heard it, resolution, capture efficiency, transcriptome coverage, that are all the things that have to be improved in the future. You need to optimize the protocol for each tissue, but probably in the future there will be a lot of protocols out there that make lives easier of the scientists. And spatial comes along with quite high costs. So one sample is around about 1,500 euros so far without the sequencing. And then of course you need sophisticated methods to analyze all the data you are generating. Has it the potential for new biomarker discoveries? I would say yes, but it's not a standalone technique. So we always need to combine it with single cell or also with immunofluorescence, in situ stainings, and so on. It's not a standalone technique, but it's definitely an asset to understand the local microenvironment and also an asset to understand cellular communication in the tissue. With this, I would like to end and thank all the people that were contributing to this work, where I have presented you the data. And I guess it was worth crowning this method of the year 2020. And I'm really looking forward to the future and looking forward to skin research using spatial transcriptomics.
Hi, Stephanie. Thank you. That was a beautiful talk and beautiful work, and I'm hoping it gets in revision. So I think you've also run into similar issues we've seen with capture and efficiency. How much tissue optimization did you do? I think other groups have tried to maybe increase the tissue thickness sectioning to get more transcripts down. I could see diffusion being a bigger issue in that respect,
Yeah.
but did you play around with that at all? I'm curious.
Yeah, we played around, and we had the feeling that enhancing the tissue thickness does not really improve the capturing, because by this you also have issues with the permeabilization in the end. Yeah? So we stick to 10 micrometer sections, and that was giving us really nice results. Yeah, but you're definitely right, and I'm waiting for all the protocols that will come out when we are really going to the resolution of just 2 micrometers, because then it's really, really tricky to make sure that all the mRNAs go exactly down there where they should go, so that the thing can be barcoded properly.
Beautiful talk, thank you. So, given your interest in kind of cell interactions and the composition of your signatures, I think this is probably a question that will be answered once we've got better spatial resolution. But I'm putting it out just for kind of interest. So, if you think about a cytokine, let's say IL-13, it's likely to have a different transcriptional profile depending on which cell type it's interacting with. And yet we kind of use these signatures that are derived from single cell types as the IL-13 signature. So I didn't know if you'd looked particularly in the dermis to see whether there was a kind of a profile dependent on what cell you were guessing was in your spot. So in other words, did you see a different IL-13 signature in fibroblasts compared to dendritic cells? Because I suspect that's one thing we don't know much about.
Yeah, actually, that's one of the weak points of our study so far, because we just were able to get keratinocyte response signatures, because we stimulated just keratinocytes with our recombinant cytokines. But that's, with the fibroblasts, I completely agree, and thanks also to the talk of Helen. We know that they are important, and I guess that emphasized it even more. So we need to investigate the function of cytokines in the dermis as well. And this would be actually one of the natural next steps in our study as well. Yeah, but it's more difficult to get a clear-cut fibroblast signature. So what is actually IL-13 inducing in the fibroblasts? So we need the data sets to understand that in more detail.
So beautiful talk. Quick question. Now we have IL-13 antagonists versus dupilumab that antagonizes both IL-4 and IL-13. Did you look at the signature of IL-4 and also IL-22 that seem to potentially also be important in atopic dermatitis?
You mean IL-32?
No, IL-22 and IL-4.
No, we did not. So we, of course, had a look on IL-22 distribution in the spatial data set, but we did not perform, or we did not create a response signature so far for IL-22, because this you can't do in 2D cultures. So if you stimulate 2D cultures with IL-22, not much is happening. So that's why this is something that we would like to do in the future, but with stimulated 3D keratinocyte models to understand really the IL-22 signature in more or less real keratinocytes epidermis.
And IL-4?
You mean co-combination of IL-4 and IL-22?
No. You showed IL-13, but what about the other cytokine that may be important in atopic dermatitis, IL-4? There are signatures out there of IL-4.
Yeah, there are signatures out there, but we could not find IL-4 transcripts in our data set. So that's why we had to focus on IL-13, because IL-4 was simply not there.
Got it. Thank you. No, beautiful work.
Okay, well, thank you very much. Really great talk.
Thank you.
So finally, it gives me great pleasure to introduce Emma Gutman, who needs really no introduction. She's well known to everybody, I'm sure, in this room, but just to give a brief resume. She's co-founder of the IEC and is the immediate past president. She's Systems Chair and Warburton Professor of Dermatology and Immunology at Icahn School of Medicine at Mount Sinai. She's won many awards, made major contributions to understanding the immunology underlying eczema and other disorders such as alopecia areata, and it communicates very well with the translational aspects that then come forth in terms of the therapeutics. She's won a number of prizes and published more than 275 publications. So we welcome you to this session, Emma.
Thank you so much for the invitation to co-chair and give a talk here. Let me just see that I'm able to get my talk. Okay. So I'm very interested also in the techniques you heard, spatial transcriptomics, single cell, but I'm going to talk about something that became my personal passion in the last few years, defining minimally invasive methods to quantify and phenotype atopic dermatitis in both adults and children. My disclosures, and Mark Lebel, my previous chair, once said that if you have many disclosures, then it means you don't have any disclosure, and I'm a total believer in that. I don't have any financial gain from any product, but I'm actually quite fortunate to have been a major part, I think, in the therapeutic development that is ongoing in atopic dermatitis and now also in alopecia areata. So atopic dermatitis for many years did not have a good treatment, as we know. Not topical treatments, not systemic treatments. And when I say good treatments, maybe I should say specific treatments, because yes, we didn't have a cyclosporin, methotrexate, phototherapy, but all these treatments, first of all, are non-specific and they harbor either side effects or they are not sustainable for long-term use. So we needed better treatments for long-term use that would be specific to atopic dermatitis. And what was the issue? The issue was that atopic dermatitis is this very heterogeneous disease on one hand, and quite complex because it has immune abnormalities, it has barrier abnormalities, and it has microbiome abnormalities, and we know that the itch is a major component. So pharma companies were confused, really, what they should go after. For quite some years, they heard, oh, you need to go after the barrier, let's replenish filaggrin and it all will be over. We know that that's not the case, but when I started my work, people told me it will be a dead end for my career because atopic dermatitis is finished. It's filaggrin and that's it. We know that that's not the case today. So atopic dermatitis has immune abnormalities with upregulation of Th2 and Th22, IL-4, IL-13 are involved, also IL-31, IL-22. So several immune axes. When you are in chronic stage, also Th1 is important with interferon gamma upregulation. And there is a major barrier defect also in atopic dermatitis with downregulation of filaggrin, loricrin, so terminal differentiation proteins, lipids, claudins, and tight junctions. And this allows penetration of irritants, allergens, and microbes, and all of this creates this complex phenotype of atopic dermatitis. So how did we get to this point in atopic dermatitis? So I was fortunate to do my postdoc in the lab of Jim Kruger at Rockefeller University that I think was very instrumental in identifying the pathogenesis of psoriasis. And I think we are going through the same path in atopic dermatitis. So this path involves, it's not an easy path, and it took some time. It involves bench studies of pathogenesis that needed to have skin and blood profiling that helped us formulate pathogenic hypotheses. Some were right, some were wrong. How did we understand if they were right or wrong? Through clinical trials with targeted agents. And it's very important to do that because while work in mice is important, I have to tell you that 95% of these studies at the end did not prove true in humans. So it's very important for those of you that do work in mice to also collaborate with people that do human work to really understand if this holds true in humans. So this also included skin and blood profiling generating biomarkers of therapeutic response. And, it wasn't easy. For each success story, I would say there were 9 failures, but these failures are also important. And generally, like I tell my mentees, in life, you need both successes and failures. Sometimes the failures may be more important, in fact, to frame pathogenic concepts and therapeutic directions. And in 2012, with a very successful fellow at the time that I'm very proud of her, became an academic pediatric dermatologist, Julia Gittler, we put forward this idea of the paradigm shift we are experiencing in the pathogenesis of atopic dermatitis. And what we said is that it's basically a futile discussion what starts first, if it's the barrier or the immune abnormalities, because what's important is that the immune abnormalities are perpetuating the phenotype of atopic dermatitis through chronic stage. So already the non-lesional skin of atopic dermatitis patients is abnormal. There is slight upregulation of inflammatory markers, including Th2 and Th22. This intensifies in acute disease and even more in chronic stage. And you heard a lot about IL-4 and IL-13. They have important functions on the barrier of patients with atopic dermatitis. They disrupt filaggrin, loricrin, involucrin, and lipids. They also inhibit antimicrobial peptides. That's important because patients with atopic dermatitis have staph colonization and infection. IL-31, the itch cytokine, right, starts the itch scratch-like unification cycle. We know that itch is a hallmark of atopic dermatitis, and this is the first symptom that we want to alleviate in patients. IL-22 starts the onset of hyperplasia, that regenerative epidermal growth, and together with IL-17 synergizes to induce the S100s, and also was shown to disrupt the barrier with disruption of filaggrin, loricrin, and other barrier molecules. And all of this, again, is perpetuating towards chronic stage. And you recognize many of these molecules, and of course there are more today, being targeted successfully in clinical trials or already approved and are in the market. Now, a very important question, and this has been a large part of our research in the lab, is the question of, is atopic dermatitis a single disease, or it's very heterogeneous and we need to target with different cytokine targeting? We need to remember psoriasis is really more homogeneous, I would say, not completely homogeneous, but it's really focused on IL-17, IL-23 biology. Whereas in atopic dermatitis, we showed multiple differences depending on age. For example, children have a different distribution, we know, clinically. But also this translates, when you look under the microscope, to differences in epidermal barrier molecules and differences in immunopolarization. They have increased Th17 activation. They do not have Th1 activation like adults. Also based on ethnicity. For example, African Americans or Asians with AD, this is a collaboration done with Kenji Kabashima. So Asians with AD have Th17 activation much higher than European Americans, and African Americans have only Th2 and Th22 activation. They do not have a Th1 or Th17, very small portion of Th17 activation. And this translates also to barrier abnormalities. For example, African Americans almost have no filaggrin abnormalities, unlike European Americans. Now, it's important to still remember all the atopic dermatitis subtypes are sharing that robust Th2 activation that we just heard about, but we need to stratify biomarkers specific to different atopic dermatitis phenotypes. And this is important in order to really develop what we hope to be a personalized medicine approach in atopic dermatitis, similar to what we have now in asthma or in cancer. So how do we get to this development of a successful translational medicine approach in atopic dermatitis? So few concepts that are important for you to remember. Unlike psoriasis, even the best clinician cannot really quantify fully atopic dermatitis clinically. Pay attention to the very high placebo responses. Some studies just presented at the ADV had placebo responses over 40%, imagine. When in psoriasis, we are talking about placebo responses around 5%. Because, right, you know, the lesions are very demarcated. You know where they start, where they end. And defining atopic dermatitis specific biomarkers in both skin and blood can help us first identify a pathogenesis or pathogenic pathways. but also will help with a development of precision medicine approaches for atopic dermatitis. Now, currently, as you heard, skin profiling is considered the gold standard to investigate biomarkers associated with a severity of the disease. But we shouldn't completely forget about blood, because it's easy, right? It's easy, it's accessible, may still be useful in order to stratify patients like it's being done now for cancer or for asthma. And we need to remember, we need to develop more non-invasive, or it's hard to call it completely non-invasive, but let's say minimally invasive skin biomarkers, such as tape strips or tiny, tiny biopsies in both adults, but particularly in the population that is so affected by atopic dermatitis, right? Children. This is the majority of our population. We need to remember that. And in that population, I challenge you to ask parents to obtain skin biopsies. I know in my collaboration with Amy Paller, she did the unbelievable, and she managed to get skin biopsies. I cannot do that in my practice, I can tell you. She probably has special talents. And defining biomarkers in specific atopic dermatitis phenotypes based on age and ethnicity and others can assist us in the therapeutic selection and trial design for our patients with atopic dermatitis, both in adults and children. Another important concept is the idea that, very similar to psoriasis, atopic dermatitis emerges to be a systemic disease. What does it mean, systemic disease? And we are talking, of course, about moderate to severe patients. We showed that patients with mild disease do not have systemic disease. So we did several studies in which we compared patients with atopic dermatitis with moderate to severe disease, not only to controls, but also to psoriasis patients. And we showed systemic immune activation when you look at activated T cells and circulatory cytokines. And importantly, when you look throughout the ages, from infants with AD up to the age of 5 with recent onset atopic dermatitis, then children 6 to 11, this is work we've done with Amy Paller, 12 to 17, so adolescents and adults, we saw that in all of them, IL-13 producing cells that are homing to skin, that have CLA positivity, cutaneous lymphocyte antigen positivity, are shared among all of them. So very early in life, already in infants, you already have increases of these cells in the circulation, but there are some subsets of cells that are different. Pay attention, for example, to the IL-9. So IL-9 that is associated with allergic responses and with asthma is mostly increased in children and adolescents, whereas you see this increase throughout life of IL-22 producing cells and interferon gamma producing cells towards disease chronicity in adults. Now, a large study we've done with Patrick Brunner that phenotyped patients with moderate to severe disease as compared to controls utilizing the Olink proteomic technology that we adapted now in many studies. You can pick multiple sets of markers. He used 350 markers. And pay attention that we have increases in atopic dermatitis patients, not only in Th2-associated markers, but also Th17, Th22, Th1, and importantly, also atherosclerosis-linked markers. And in a recent study that, again, looked at Olink proteomics, but at multiple ages, not only in adults, but also infants, children, adolescents, and adults as compared to their healthy controls, so relevant controls, we saw that in the infant population, you have specific increases of different markers that are linked to IL-17 and other markers. But when you look at the prototype of Th2 and Th22 and Th1 associated markers, you see this gradual increase from infancy towards adults of markers like CCL17, OX40, and others. Now, importantly, when we think about minimally invasive techniques, we need again to think how we minimize, on one hand, skin biopsy, or we are developing additional techniques. And we utilized, again, Olink proteomics that allows to use in tissues only 10 micrograms, which is equivalent to only 1 millimeter punch biopsy. Very, very tiny punch biopsy. In the United States, you don't need to close it with a suture. I know in Europe and Israel, for example, you don't need to close with a suture a biopsies up to 4 millimeters, but in the United States we do. So these biopsies you do not need to close with a suture, and in these biopsies that we've done in patients with atopic dermatitis in adults, with moderate to severe atopic dermatitis, we performed all-in proteomics, again, 350 proteins, and we obtained between lesional, non-lesional, and normal 161 differentially expressed proteins. Instead of DEGs, think DEPs in lesional skin and 69 in non-lesional skin, as you see here. When we zoom into that, we saw that many of them were not only just inflammatory markers, such as a CCL17 that we found and others, but also markers associated with atherosclerosis, such as selectin E and others. And when we look at pathway analysis, we found that atherosclerosis signaling was highly upregulated in lesional skin and non-lesional skin as compared to controls in the skin. This is in the skin. And we compared the skin phenotype of these patients to the blood phenotype of these patients. And here the circles are basically compatible with higher fold changes of upregulation compared to normal. And what you can see clearly that when you look in the skin, the expression levels were much, much higher than in blood. So these findings are suggestive likely that the skin is likely the source for the inflammatory and cardiovascular risk proteins that we see in these patients. So proteomic profiling may be very valuable for studies that require biomarker monitoring, since the all-in platform requires very little, both in blood and in skin. In blood, we need only 10 microliters, and in skin, very little. I told you 1 millimeter punch biopsy and 10 micrograms. So to put it together, I think we need an integrated approach when we consider biomarkers. We need to consider the lesional skin, we need to consider the non-lesional skin of patients, and of course we need to consider blood. So when I'm consulted about biomarker approaches, I always say that we should consider everything, and then we need to see what correlates with the severity. So now let's switch gears and ask, how are we testing the contribution of the different cytokines and immune axis to atopic dermatitis? I told you that clinical trials are instrumental to understand what happens when you shut off one pathway. And of course, these are best when coupled with biomarkers. And dupilumab was really instrumental, not only for becoming the first specific treatment going into atopic dermatitis, but also for dissecting the pathogenesis of atopic dermatitis. Again, it targets IL-4, IL-13, because it targets the receptor. And IL-4 and IL-13 were showed by multiple studies to be involved in the barrier disruption in atopic dermatitis and the lipid disruption as well in atopic dermatitis. And in this study that we performed, we started with 54 patients. This was the mechanistic phase 2 study of dupilumab, so it was a relatively large study, 54 patients, 27 on drug, 27 on placebo. It was with weekly dupilumab, but dupilumab 200 milligram that was unique to this study. And this is the RNA-seq data, and you can see red is upregulation, blue is downregulation. You can see in lesional baseline a similar upregulation of markers, but only with drug and not with placebo, you see that it turns blue. So there is a marked downregulation of multiple markers. What markers are primarily downregulated? These are Th2. Markers such as CCL17, CCL26, you heard a lot about them already, CCL18. Pay attention that dupilumab doesn't do much to Th1. Interferon gamma, CXCL9 remained not touched, but they were not also significantly increased. And this is important because when we have facial rashes, sometimes it is contact dermatitis that involves the Th1 axis. But the Th17 axis was actually downregulated with dupilumab. Now, I think the million-dollar question is, what happens with the barrier? Is dupilumab able to reverse the barrier? The answer was yes. You see that lichenification at baseline, it completely shuts off the lichenification. And when you look at the epidermal thickness of the entire patient population, 40% decrease. The keratin 16, that is a good proliferation marker, is completely shut off with dupilumab. and what happens with filaggrin. Are we able to normalize filaggrin to really prove that immune hypothesis? And the answer is yes. Pay attention to the skipped expression of filaggrin and the normalized nice line of expression after treatment. So I think this study was really instrumental in showing that dupilumab impacted not only the inflammation, like one may think, but also the barrier dysfunction of atopic dermatitis. And this was the first study that managed to establish or to prove that the Th2 axis and IL-4 and IL-13 cytokines are pathogenic in atopic dermatitis, cementing AD as both reversible and immune-driven like psoriasis, and frankly, opening the door to all the therapeutic development that is ongoing now, because before, companies did not want to touch atopic dermatitis. I heard when I started my career from so many companies that atopic dermatitis is a barrier disease and they don't want to go to that disease, and now see what success story it is. Now, dupilumab also inhibits systemic type 2 inflammation. In the same study, we showed CCL17, that's TARC, and CCL18 to be significantly downregulated with drug, not with placebo. And pay attention that also you should use systemic biomarkers to complement your studies. Now, this is my own NIH study with fezacizumab. Fezacizumab is an anti-IL-22 antagonist that Pfizer had on the shelf after it was not successful in 2 studies, in psoriasis on one hand and RA. So we took a drug that was not successful, but we took it to prove, and we are very grateful that we had that opportunity to prove the potential pathogenic effect of IL-22 in atopic dermatitis. And IL-22, we and others showed that it's important for the hyperplasia in atopic dermatitis and in the barrier defects in atopic dermatitis. This was a very small study. Today, studies are much larger, only 60 patients, 2-to-1 to placebo. We unfortunately placed the primary endpoint at week 12. Today we would have put it where the secondary endpoint was at week 20 because it's a slow drug, but still you can see differentiation between drug and placebo. At week 12, only it was not significant, but when we did a regression model that took severity into account, we did obtain significance at every single week. So then we decided to divide patients to moderate, these are the non-severe because the study enrolled only moderate and up, and severe patients who scored more than 50 and up. And here you see actually significance at every single week. So we asked ourselves, what does it mean that the more severe patients responded more? So I had a crazy idea to divide patients based on the median baseline IL-22 expression, and this is a heat map of RNA-seq that we've done. And basically, I consider this the first example of personalized medicine because the patients with high baseline IL-22 improved quite well, and this is a monotherapy study. Red is turning into blue already at week 12. The biopsies were at week 12, not week 20. And basically, the lesional skin looks very similar to non-lesional skin. You don't see that in placebo where red is staying red. But what happens to low baseline IL-22? Unfortunately, we made these patients worse. Blue turned into red, so they exacerbated. So no improvement, even exacerbation. So for those patients, and it was about half of the patients, we really helped them. And not only we held them, and that was not one of the endpoints, but we've seen many patients like this with highly lichenified lesions, very hard to reverse such lesions, staying clear for several months after. So potential for disease modification. And we know that now there are companies targeting IL-22, so we should follow this because I think this may be an interesting target for the next few years. So now let's talk about our work in tape strips and development of a less invasive approach in atopic dermatitis. So let's review some principles of tape strips. It was introduced as a detection method of melanoma because pagetoid melanocytes are really superficial, and it was possible to capture them. And we have adapted this technique to study inflammatory skin diseases. It removes the stratum corneum, and Donald Leung showed that it removes it up to the stratum granulosum. And the benefits, non-invasive is maybe a little bit too stretched. I would say minimally invasive, but it does leave no scars, and that's important. And it is relatively painless, or even if there is a little bit pain, it goes away quite quickly. So this is the technique. These are the tape strips we are using on the lesion, either 15 or 20 times. This is the pressurizer. And in this study, we had atopic dermatitis and healthy control skin of different age groups. The pediatric ones are done together with emipaller, so infants, children, adolescents, and adults. And before I showed you that, I want to show you our study in adults that was together with Robert Bissonnet, adults with atopic dermatitis and psoriasis, as compared to normals. And Helen, that is here, is the first author, and will have a follow-up paper to the study. But in this study, we wanted to see if we compare psoriasis, atopic dermatitis, and normal, if we are able to identify a profile that is similar to the profile we identified in biopsies. And we've done RNA-seq, and imagine the tiny quantities that you have. And we were surprised to have so many differentially expressed genes in both diseases, in atopic dermatitis and psoriasis, more than 4,000 in AD, and more than 5,000, believe it or not, in psoriasis compared to controls, using the same criteria. Fold changes more than 2, and fast discovery rate more than less than 0.05. And when you zoom into the differences between these diseases, because you want to see that you maintain the same differences that you saw in biopsies, we indeed saw that. So in atopic dermatitis, as expected, we found CCL17, right, one of the hallmark biomarkers of AD. Pay attention to the high fold changes of expression in AD. CCL22, this is OX40, TNFR, SF4. Pay attention to the high upregulation in atopic dermatitis, not so much in psoriasis. Remember, it had a failed study in psoriasis, no wonder. But now we know that it's highly expressed in atopic dermatitis. IL-13 is there, and CCR4, another target that is going into atopic dermatitis. What's happening on the other hand? In psoriasis, we have the genes that we know to find in psoriasis, IL-17A, IL-17F, NOS2, IL-17C, and some Th1 markers upregulated in psoriasis. And next, we wanted to see if we can define, based on tape strip, a molecular classifier differentiating between psoriasis and atopic dermatitis, and we patented this because using tape strips, we managed to get only one biomarker alone. In biopsies, it was shown that you need 2 biomarkers. However, in tape strips, you only need one biomarker that 100% certainty is identifying if your lesion is psoriasis or atopic dermatitis. Perfect classifier based on tape strips. Next, we moved on to see if, compared to biopsies, we can identify the profile of babies with atopic dermatitis or infants between 6 months old and 5 years old. And this is work that we've done with emipaller. Children with really very early onset atopic dermatitis. They needed to have atopic dermatitis within 6 months of initiation when we tape strip them. And here are heat maps of RNA-seq we've done, and we separated these so that you can see it here for immune genes, differentiating the lesional skin of these babies, non-lesional skin, and normal, and barrier genes that were previously identified by us in the past. And I want to point out CCL17 is here. You have IL-13, also OX40 in this list, and barrier genes, many of them are there. Pay attention to filaggrin, highly downregulated already in early life. Importantly, a biomarker is not really a biomarker if it doesn't associate with severity. And here we try to associate biomarkers with severity, both with EASI, but also pruritus, transepidermal water loss, not only for lesional skin, but also non-lesional skin. Pay attention to IL-4 receptor, IL-34, a negative regulator, and filaggrin. and ICOS, that is a marker of T-cell activation, and also non-lesional skin, again, filaggrin, IL-34, and MMP12. And more recent work, recently published by Yael Reinert when she worked with us in New York City. Now she returned to Israel. I'm very proud of her. She's now a pediatric dermatologist at Beilinson Hospital in Israel. So here we wanted to see how the phenotype differs among infants, children, and adolescents based on tape strips. And pay attention that the same markers I showed you based on biopsies, you also can see them here based on tape strips. Important to remember that some barrier genes such as filaggrin and loricrin are showing greater dysregulation, so basically they are more downregulated later in life in children and adolescents, not so much in infants. But, for example, lipids are actually more dysregulated in infants, whereas important hallmark genes that identify the inflammation in atopic dermatitis, such as the Th2 inflammation like CCL17 and CCL18, or the Th22 inflammation S100s, are similarly upregulated in all of these age groups. And this is a word cloud representation that represents the fold change differences compared to controls from non-lesional skin. So this is non-lesional skin. And remember the very nice work just presented at the ADV by Jacob Tyson's group, showing that CCL17 may be a predictive biomarker of a development of atopic dermatitis. And it's not surprising that in infants, it is here, and Jacob and I will collaborate on some tape strip data also from his cohort. So super exciting times in atopic dermatitis. Pay attention also to the barrier genes that already are expressed very early in life, and F828, that is a lipid that is highly dysregulated very early in life. So now I think we need to end by asking, can tape strips be used to evaluate biomarkers of therapeutic response in atopic dermatitis? Because while techniques like spatial transcriptomics, single cell, and even just RNA-seq or immunohistochemistry are really exciting, you cannot do that in all the patients in a clinical trial. Simply not feasible. You need to find some methods that you'll capture all the patients in a clinical trial. And the answer is definite yes. So this is a study we've done with dupilumab in real life. So we gave dupilumab in real life to 26 patients, and we evaluated pre versus post. And this is a heat map of RNA-seq we've done. And pay attention that many of the markers we've seen in the biopsy analysis were here too. MMP12, marker of general inflammation. We have some Th17 markers like IL-19 that we heard, T-cell activation markers such as ICOS, terminal differentiation markers, highly represented here, increased with dupilumab, such as filaggrin, dendritic cell markers, and importantly, the Th2 markers that are the hallmark of dupilumab, right? And pay attention that OX40 is again here, so dupilumab downregulates OX40, and CCL17, of course, is here as well. Oh, sorry. Now, next, we wanted to understand if tape strips represent well compared to biopsies, what happened after treatment. So this is a comparison not done at the same time, and it's not the same patient. So we took our study with dupilumab, the clinical trial data, and compared the same biomarkers in this data set. So pay attention, and it's not the same patients, but you can clearly see that the results are in the same line in tape strips and in biopsies. Here I showed you only Th2, but you need to believe me that the same holds true for all the other markers. So Th2, very similarly downregulated in biopsies and tape strips. Again, when you see biomarker studies, don't believe these if you don't see correlations with clinical data. Biomarkers have to correlate with clinical data, and this is the easy improvement in the study, the real-life study, 70%. Funny, it's the same improvement that was in the clinical trial. And here, many biomarkers correlated with the clinical results and also with transepidermal water loss. And we also, from the same study, we managed to also perform proteomic analysis using Olings on tape strips. using only 2 tape strips, and this is the data. Also, Helen is the first author in this study. And we see biomarkers like CCL17 and CCL13 highly downregulated in proteins. Only 2 tapes were used for the study. So I think we have a super exciting times in atopic dermatitis. I hope all of you enjoyed the symposium, and I'm really excited for the years to come. Thank you so much.
Thank you very much, Emma. Any questions? So maybe I could start then with asking, in terms of ongoing clinical trials, how do pharma regard non-invasive tape stripping to run in parallel with the more conventional techniques? What's their view on it?
Yeah, so I think I'm aware of several studies. In fact, we collaborate on several studies that ongoing efforts on tape strips are happening. I think tape strips have a plus and a minus. The plus is that it's non-invasive and more patients can do it, and definitely children, you can do it. The minus is you cannot do it in hair-bearing areas. So, for example, it cannot be done in alopecia areata. Very important to remember that. It's hard to pull. And secondly, it necessitates, it's not a minus, but it necessitates a lot of lab resources and lab work for the extraction of the tissues. But I think it's a very valuable approach, and we are now using it quite often. I mean, we are using also the other techniques that I think are very valuable, but the other techniques, for example, spatial transcriptomics and single cell, I cannot see being done in a lot of patients in a study just because of cost and so on. So I think it's important to think about different techniques for your purposes.
Okay, yeah.
So recently I saw some papers that they did suction blistering and in single cell analysis, and they found like a good correlation between other invasive techniques. Have you tried to apply it or?
Yeah, that's an excellent question. So I think suction blistering works well. From my experience, you know, I'm in New York City, and patients will not stay several hours. The patients need, we actually tried that. I think it's good to get information out of it, but the patient needs to stay 3-4 hours to have the suction blister, whereas the other techniques, they are fast, and frankly, I think you get as much, if not more. So I don't see the added value.
And how do you see the technology affecting the size of biopsies? Because I think that certainly is a factor, isn't it? That persuading patients to consent to giving skin for clinical trials is somewhat challenging. Although when we've done it, I've always been pleasantly surprised how the patients actually want to give something back to the clinic because they've taken a lot. But I think if we could hone the technologies down such we were doing 2mm punches rather than 4 or 5, I think that would be a major advance. But I don't think we've quite got there yet. But what's your view?
Yeah, that's an excellent question. And funny, I had this discussion with Jim Kruger. For those of you that do not know, he's my husband. And he, in psoriasis, was doing 6mm punch biopsies. And when I started, I was like, oh, this will never fly in atopic dermatitis. And slowly, I minimized it. And it depends what you want to achieve. I think if you want to achieve only RNA-seq, you can do that only in 2mm punches. But if you want to achieve also immunohistochemistry, immunofluorescence, or to consider doing spatial transcriptomics, you have to do larger biopsies because you need to cut the samples. There is no way you can cut a sample that is, let's say, 3.5, you will not succeed very well. I would say 4mm. So it depends. Right now, we improved our technique. So from 4.5mm biopsies, we are doing everything. single-cell spatial transcriptomics, RNA-seq, and immunohistochemistry.
Any final questions? If we could please pass the microphone back, that would be great.
Thank you for your interesting talk. If I got it correctly from the talk from Dr. He, she said that CCL17 is mainly produced by fibroblasts, actually. So can you comment on how this is actually a potential biomarker then for treatment response?
That was CCL19.
Oh, it was 19. Thank you. Good.
Okay, well, I think it's been a really fantastic start to the ESDR, and thank you all for coming, and a particular thank to our excellent speakers who've set the scene, and we look forward to interacting at further IEC meetings as well as continuing during the ESDR.
Yeah, thank you so much. And thank you. And thank you for the speakers, amazing speakers. I learned a lot today.